the classical virology, we have been just, in order to identify and to get sequence of the virus, we found sick animals. By sick animals, we found animals that they have tumors, and then we directly extract a tissue samples from the tumor, and from there we just use the normal DNA extraction process, or RNA if we're working with it, and just trying to identify it. The curious part about my project is since we were interested in analyze worldwide turtles from everywhere, so we got tumors only for 3 different populations that were the animals that we knew they were sick. And we got samples, just normal tissue samples from animals everywhere else. I did the sampling for several populations. I was in the Middle East, in Kuwait, Oman, and Dubai, and they have a small population of sea turtles there. And when they come to nest, when they come to land to lay the eggs, simply we just took a small piece of tissue from the flippers, and that was it. We thought that we were using those animals as a negative control because the animals had never been reported, they didn't show any sign of the disease. And the same thing was done for many other samples from the Caribbean, from Portugal, from basically everywhere else. And the interesting part is that we start finding that those animals that didn't have any sign of the disease or they had never been reported, they were positive with the virus. And that was when the whole project started becoming more interesting, because it's like, okay, what we thought there was something very random, very unique for some only unique populations, is actually not. It means like, and just imagine if for humans you could, with the SARS example that you just put, that you can really track how the disease got spread around the world. Sea turtles are migratory animals. They move around the world following currents, and it's really, really, really difficult to track where is the migration of those animals. And when you start comparing sequences from animals sick in Hawaii and compare this one with from the Caribbean, and you compare that those 2 sequences are almost identical, so you start wondering, okay, how this spread occurred? It cannot just be something that just randomly occurred and then by any chance the animals from Hawaii got infected from animals from the Caribbean. So there is something that you need to go back in time in order to understand that.